So hi and welcome everybody. Welcome to you who are here in Zoom. And if you're following us on YouTube, well, welcome to this FibRe webinar. This time we have Professor Kevin Edgar from Virginia Tech as a presenter, and we will hear more of, we'll hear a presentation soon. Just one thing about questions. If you have questions to a speaker, we will have a Q&A session after the presentation. So if you are in Zoom, you can raise your hand or type questions in the chat. And if you're on YouTube, you can use the chat function. So to give our speaker the best introduction, we now have Professor Ulrika Edlund, who will give an introduction. So please, Ulrika.
Thank you.
I think I'm the one who will give it. So,
okay, sorry. Then of course we have Professor Annette Larsson instead given an proper introduction.
I will try to do my best.
Yes.
So it's a great pleasure that we have Kevin here. Professor Kevin Edgar is located at the Department of Sustainable Biomaterials at Virginia Tech. Professor Edgar grew up close to Manhattan, less than 20 kilometers from Hudson River. And he got his PhD in organic chemistry from Duke University. And he has also experience from industry as Eastman Chemical Company. His research focus on synthesis of sustainable polysaccharides and do it in a sustainable way. And this is done to aim for replacing fossil-based materials and still have very high performance. Edgar is also exploring structure property relationships to modify unmodified and unmodified polysaccharides. And with this structure property relationship knowledge, he can then apply it to different areas like drug delivery system, but also control nanostructures for other innovative application. Professor Edgar is also co-founder of the Gordon Conference for Polysaccharide Chemistry and has also introduced a model that so this conference can rotate between North America, Europe, and Asia. He is also editor-in-chief for carbohydrate polymers, which has a very good impact factor, and it has raised a lot during the last year. So it is my great pleasure to introduce and give the word to Professor Edgar.
Thank you so much, Annette. It's a great pleasure to be able to talk to this group. And you made the mistake of giving me quite a bit of time. So I'm actually going to talk about these topics and one more, if I can actually get my slides to move. Come on, slides. There we go. And it's not a surprise to you that we really focus on these wonderful polymers we call polysaccharides that are so abundant in nature. They're so diverse in nature. They're always biodegradable. They're renewable, and they're, we think, underutilized. And so we're going to talk about three different broad areas, one about sustainable plastics, one about self-healing hydrogels that are all made entirely from polysaccharides, and another about enhancing solubility, particularly solubility of drugs. So let's start talking about plastics and Graft copolymers and how they can have a positive impact on plastics. We all understand about the problem of plastic pollution. We've got birds that are ingesting too much plastic and starving to death. We've got various creatures that are getting hung up in six-pack rings. We've got gyres in the Pacific. We've got microplastics everywhere, including in animals and in us. And so we need to do something about that. Of course, we have non-renewable raw materials that are going into those plastics, and that also is something that has to concern us. So how do we solve that? Obviously, one of the ways we'd like to solve it is by turning towards polysaccharides. Now, of course, there are polysaccharides in plastics today. There are wonderful cellulose ester-based plastics. Could be that your eyeglass frames that you're wearing right now might be made out of cellulose acetate, or the the bottle opener or the hammer or the screwdriver that you used last night might have had a cellulosic handle, or the Scotch tape that you use, of course, is made out of cellulose acetate. So there are great plastics, but what about degradability? What about renewability? Well, back in the 90s, actually, Charles Buchanan at Eastman showed that cellulose acetate is biodegradable. That mattered because up until then, and even up to this day, people that aren't aware of his literature think of cellulose acetate as not being biodegradable. Primary reason for that being the fact that cigarette butts are made out of cellulose acetate, and we see them stay in the street, in the gutter, for a long period of time. And people thought that was because cellulose acetate was not biodegradable. It's not. That's not the reason. The reason probably is the nicotine. But Charles did this wonderful experiment of radio labeling cellulose acetate at the ester group, and then putting it into a composting environment and following labeled carbon dioxide that was evolved. And what you can see is that this was for several cellulose esters, so you can't really see, but the cellulose acetate propionates, both of them are down here on the bottom. So they're, over the course of a few weeks, they're not biodegrading. But then you get up to DS 2.5 or so, which is roughly that same DS that's used for cigarette butts and for tool handles and eyeglass frames. Does biodegrades quite nicely over the course of about 3 weeks. And a lower DS, cellulose acetate 2.05 or so, biodegrades even faster. And he also identified that DS 2.05 material was an excellent plastic. It had excellent plastic properties, right? So there is a way to deal with this for certain plastic objects. We can ask, though, what about starch? So for many things, your soda bottles, your milk bottles, we want them to last quite a while, right? We don't want them falling apart on us. We certainly don't want them falling apart in a liquid environment, because they're meant to capture liquids. So we want certain plastics to be durable, and those durable plastics, the ideal thing would be to recycle them. But there are lots of single-use plastics. We can think about the concept that I think was also very popular in Europe of taking a fast food restaurant and making sure that all the plastics that are used in that fast food restaurant, the cutlery, the glasses, the dishes, everything is made of a biodegradable plastic, such that the garbage from that restaurant can be taken straight to a composter, can be turned into valuable soil, right? And so there's not going to be any waste that comes from that restaurant. Everything that goes into that restaurant is going to come out as something useful and helpful. So we'd like to have a plastic that works for such single-use applications. You could also think about agricultural film. You could think about diapers, right? There are a lot of things that you don't want to recycle. You want them to be biodegradable. So what about starch? Great idea, right? It's abundant, it's biodegradable, has this wonderful process that I don't know of any other polysaccharide about which this is true. We can thermally process it. Like all other polysaccharides, its glass transition and its melting temperatures cannot be observed, right? Because they degrade before they soften or they melt. As we know, polysaccharides don't degrade at particularly low temperatures. What they do is have TGs and TMs that are so high that they can't be observed. They're above the degradation temperature, right? So the thing about starch is it's plasticized very well by cheap, benign water, right? So we can add a little bit of water to starch. We can thermally process it. We can make pellets or sheet or whatever we like to with it. So that's a wonderful property that starch has. That's great, but it's a bad idea because starch objects are water sensitive, right? It's not a structural polymer in nature. It's meant to store photosynthetic energy, not to have structural applications in nature. And it's not a good structural polymer. So this experiment you see in this little chart here is people made a starch tray. They put out some starch sheet. They formed it into a tray. They put water on that tray, and your yellow line here just shows the tray literally disintegrated. The time frame here is minutes, right? So over an hour to an hour and a half, the tray completely disintegrated because it's so water sensitive. which would not be a good property for use inside of a fast food restaurant, right? You don't want your tray disintegrating while you're on the way over to the trash bin. But maybe it's a good idea, right? We know we can acetylate starch. We can acetylate starch very easily, more easily than cellulose, for example. And the cool thing about that acetylated starch is it's still extrudable and it's still biodegradable at a quite respectable rate, not that much slower than starch itself, right? So, okay, we can reduce the water sensitivity of the starch by acetylating it, and it's still got all these great properties. No, it's a bad idea because starch acetate, like starch, is brittle. It has poor toughness. It's not a very good plastic. Okay, so we solve some problems. We can't solve all the problems by simply acetylating starch. So what do? And one of the things that people like to do to improve the properties of plastics is to blend them with other plastics. As we know, that's not so easy. Not too many polymers are truly miscible with each other. You know, usually your delta H can't overcome the problems with delta S and mixing two polymers together. So two polymers we'd love to be able to mix together to solve some of these problems are polylactic acid and starch. So here's starch. We know starch has two components, linear amylose, ultra branched amylopectin. Right, so we can take starch and we can turn that into starch acetate. The cool thing is, of course, that PLA comes from starch. Right, we can hydrolyze starch down to glucose. We can ferment that glucose to lactic acid. We can combine two lactic acids to lactide and polymerize that to polylactic acid. So from renewable starch come both starch acetate and PLA. That's nice. So now they're both biodegradable, they're both renewable. But as we can see, if we blend them together, what we get is large domains of PLA in the starch in this particular case. They don't blend. They're not miscible with each other. And you can buy starch acetate PLA objects, right? They're not transparent and they're not very strong. They're only restricted to uses where they don't have to be very strong. So the blend is not going to help us much in terms of properties. What can we do about that? We want to blend these two polymers. They're both renewable. They're both biodegradable. The PLA we think could help with the water sensitivity of the starch. So what do? The idea that we had was to make a compatibilizer, to make a block copolymer from a backbone of amylose in this case, and side chains of PLA, and that block copolymer might be a good compatibilizer. And furthermore, we had a hypothesis that what you need in a good compatibilizer here is relatively infrequent PLA branches and relatively long PLA branches. And we thought that would give it the properties to mix the two phases effectively, that the branches would lie in the PLA phase, the main chain in the starch phase, and that would give us the properties we wanted. And Jeffrey Thompson is my student that's done most of this work. So how do we get there? We started with this wonderful furohydrobromination that is the most selective reaction I know of in polysaccharide chemistry. It's almost perfectly selective for brominating primary alcohols on polysaccharides. NBS triphenylphosphine brominated that. And then in the same pot, we acetylate. We acetylate all the other hydroxyl groups. And so we have essentially perfect selectivity. All the secondary alcohols are acetylated. The bromination at C6 is controlled stoichiometrically, and the ones that aren't brominated are acetylated. So the bromines are going to be where we're going to attach the PLAs. And so the frequency of the bromines is going to control the frequency of branching. And controlled conditions gave us perfect bromination at C6 and the degree of acetylation at the other positions that we desired. And it's easy to see that in the proton NMR spectrum where the C6 acetate comes a little bit further downfield than the other acetates. So we can follow the growth in that acetate peak as we go from just nearly complete bromination. That means there's no hydroxyls left to acetylate at C6 to bromination only at DS 0.04. And so this chemistry, which Jeffrey worked out, and there's a lot more details than I have time to talk about here, but it allows us to control the branching down to a very low frequency, which is the key to this whole process. So we can displace that bromide with azide. That's a quantitative reaction, right? So that azide is actually going to be the thing that we use to click on the branches. We tested out that click chemistry on this polymer with T-butyl propargyl ether. And what you can see is that the degree of substitution of that branch can be controlled all the way from a DS of 1 down to a DS of 0.04. And that the spacing between branches, therefore, can go from 0 to 20 monosaccharides. So we have quite a bit of flexibility in terms of how frequent those branches are going to be. Okay, there we go. So the next question is, we had a hypothesis that the branch length was going to be important, and how do we control that branch length, and how do we set it up to be grafted onto the polysaccharide? The way we did that was to start the polymerization of lactide with propargyl alcohol, and Jeffrey was able to work out conditions where we could really control that and get different number average molecular weight branches with great control, with very little initiation by anything other than propargyl alcohol. So now we have a branch of controlled length, and a branch that's got an entity on its end that's going to enable us to click it onto the polysaccharide. And sure enough, that click chemistry worked well, right? So we have this triazole linker between the starch, the amylose acetate, and the PLA side chain, and lots of different ways that we proved that, in fact, we had the material that we claimed we had, including DOSI spectroscopy, where you can see that we don't just have a mixture of the two polymers. We, in fact, have a block copolymer. So the real question is, what does it do? Does it actually work the way you want it to work? Remember what our hypothesis was, that we want relatively low frequency of branches, and we want long branches. I'm not going to talk about the long branch part now. That part is currently underway, but I can tell you that we were right, that we did indeed need long branches. So here we're going to look at solution cast films of this blend of two polymers that are going to either include or not include this compatibilizer, this block copolymer compatibilizer. And what we're going to see is phase separation here. This is amylose acetate mixed with PLA, right? And as we saw before, the phases separate completely, and they're just not miscible with each other. If we add a compatibilizer that's got a branch at every glucose, right, the 100% graft density, what we see is that the size of the phases is a little bit smaller, right? So we're starting to see a little compatibilization, but nothing to write home about. Here is a case where we have one branch roughly for every 3 glucoses. And you see that the size of the domains is smaller, that we're seeing some compatibilization. It's getting better. And here's where we have one branch for every 20 glucoses. And here you can see that optically, there are no phases, right? We have optically complete miscibility. Now, I'm not claiming that these two polymers are actually completely miscible with one another, but what I am claiming is that the size of the domains is so small that you can't see it optically anymore, and we're going to have much better properties from that material. So it really is a good compatibilizer, and this is one of those cases, for good or for ill, where all of our hypotheses turn out to be correct, and we can now design a very highly functional graft polymer compatibilizer. And we think this is an example of a field that's going to develop. There are a lot more opportunities in this field. So let me switch topics to the second one, which is to talk about hydrogels. This one's going to be based on selective chemistry also. It really started with this concept that my student Brittany Nichols carried out, that we could take something as inexpensive and commercially available and simple as hydroxypropyl cellulose. We could do some really simple chemistry on it, but that is designed to be selective. And so we take this Stevens oxidation that dates back for small molecules to 1980. It's been known since 1980 that you can take secondary alcohols, you can oxidize them with the same bleach that you've got in your laundry room or under your kitchen sink, household bleach, nice, dirt cheap, simple material to use. And it's going to oxidize secondary alcohols to ketones. No one had ever done this on polysaccharides before. Probably no one was ever dumb enough to try it, because you've got a lot of other hydroxy groups on these polysaccharides. Of course, a lot of other stuff could happen. But we partnered with Petra Mishnik on this. Petra broke down the polysaccharide to monosaccharides and did her thing with mass spec and other techniques. And she was able to prove that, as we thought, that it was very selective. In fact, it's 90 to 95% selective for these secondary hydroxyl termini of these oligohydroxypropyl side chains for a lot of reasons that we don't need to go into right now. So what we're able to do by this chemistry is to impart ketone functionality to polysaccharides, and that's not so easy to do. There aren't so many other ways to do that. So what? Well, the thing we thought of right away was that we might be able to make hydrogels, in fact, all polysaccharide hydrogels. And so we take these ketone groups on this oxidized hydroxypropyl cellulose that we made by bleach oxidation and react that with simple chitosan, the chitosan with its many amine groups. And they immediately react with one another, and we make a hydrogel, right? So an all polysaccharide hydrogel, no small molecule crosslinker is required. And these hydrogels have some interesting properties. So we can look at it rheologically, and what we see is that at low strain, right, we have this arrangement of G prime and G double prime. As soon as we apply some strain, they switch, right? We liquefy the hydrogel. As soon as we take that strain away, it goes right back to where it was before. And we can repeat this over and over again. And what you see with this particular hydrogel that's interesting is that the recovery is essentially 100%. It goes right back to where it was before. So that's the rheological experiment. You can also do a visual experiment. Take the hydrogel, cut it in half, put the two halves together without any pressure, and then just wait 1 minute. And after 1 minute, you can pick the hydrogel back up. It has self-healed. It's also injectable. So you can take that hydrogel, put it into a syringe, put the pressure on the plunger as though there were water in the syringe. That's all the pressure that's required. That much the interaction of the barrel and the plunger of the syringe is enough to liquefy the hydrogel. It drips out into your vial, and immediately upon completion, you can turn that vial over and it's gone back to being hydrogel. So it's injectable and self-healing. It's got some really nice properties. One thing that we saw as an issue as we're just getting into this was this odd process we had to go through to make the hydrogel. So you mix that oxidized hydroxypropyl cellulose in water with the solution of chitosan in water, but of course chitosan's not soluble. Most chitosans are not soluble in pure water. You have to add a little bit of acetic acid. So you mix those two solutions and you get this clear solution. Seems like a clear solution anyway. Low viscosity, it looks just like water. If you freeze dry that and then you put it back into water, that gives you the hydrogel, probably because of the presence of acetic acid in this initial solution. So it was a little bit annoying. It made a very nice hydrogel with all the properties that we just talked about, but the process to make them was not something we particularly liked. And so we said, okay, could we simplify that process? The hypothesis we had was the acetic acid we needed to solubilize the chitosan was catalyzing the hydrolysis of the imine crosslinks. And so we had to get rid of the acetic acid. And so what about a chitosan that's water soluble, carboxymethyl chitosan, famous for being a water soluble chitosan derivative? Of course, when you carboxymethylate chitosan, the conditions by which you do that are going to control the amount of O carboxymethylation versus N carboxymethylation. And if you carboxymethylate it nitrogen, you're taking away the amines that you need to make the gel happen. Right. So can we carry out under conditions that promote O carboxymethylation and thereby leave enough amines to actually make a hydrogel? And sure enough, we can. So that carboxymethyl chitosan made under anionic conditions is mostly O carboxymethylated. We quantified that in the paper that describes this. That carboxymethyl chitosan in water makes a hydrogel very nicely with the oxidized hydroxypropyl cellulose. Right, the gel time is related to concentration. It is a little bit slower than the gel time was for chitosan, but it's quite acceptable. And again, we have gels that we can inject that are self-healing, that are porous. They've got a lot of nice characteristics. We can also, in fact, there is the ability to take some of those ketones that we're using to make the hydrogel, to form the imine bonds that make the self-healing hydrogel, and use those to append an active molecule to the hydrogel. So we can take an active molecule, phenylalanine being the model active molecule in this case, take that amine-containing active molecule, react it with some of the ketones on the oxidized hydroxypropyl cellulose. There's still enough ketones left to make the hydrogel. And now you've appended that active molecule covalently to your hydrogel. So release of that active molecule is going to require hydrolysis of an imine bond, migration, diffusion of that active molecule through the hydrogel. And that active molecule, of course, is going to have the opportunity to react again with the ketone as it comes out of the hydrogel. So what we thought was that was going to substantially affect the release rate, and indeed it does. It does affect, it slows down the release of that active molecule. So you can imagine an injectable hydrogel containing an active molecule that you can inject into a part of the human body where you need to release a drug and active molecule over time. That's one of the ways you could use something like this. We also found that we could take those oxidized hydroxypropyl cellulose molecules and make zwitterionic derivatives by reductive amination. Right, so the last one I showed you with phenylalanine was just making imine bonds, not reducing them. So they're now hydrolyzable and they can release the active molecule. Here we're doing reductive amination with sodium cyanoborohydride. And so we're making a sigma bond between nitrogen and carbon, so that's not particularly hydrolyzable. And because we're attaching an amine containing carboxylic acid, that gives us amino acid products that are zwitterionic, that are very water soluble. We can make them from glycine to amino hexanoic acid. In other words, the monomer for nylon 6. Those all work very well. They're all water soluble, and they're interesting amorphous solid dispersion properties. And that the amorphous solid dispersion information is in the paper if you'd like to read about it. And we'll talk more about what amorphous solid dispersions are and why we care about them here in just a minute. So remember back, a couple of slides back, I said you take that aqueous solution of oxidized hydroxypropyl cellulose and the aqueous acetic acid solution of chitosan, you mix them, you get a clear solution. But was it really a solution? And we had reason to look at particle size and various materials, and we wanted to use this solution as a control for those experiments. And we were doing dynamic light scattering. We did dynamic light scattering of this alleged solution, and it's not a solution. What it actually is a nanogel dispersion. So we can take an aqueous solution of oxidized hydroxypropyl cellulose and an aqueous acetic acid solution of chitosan, just mix them together. And that gives us a dispersion of nanogels of roughly 100 nanometer diameter. And so we thought that was extremely cool and possibly extremely useful. And so we have pursued that. We thought it would be more convenient, again, to make solutions, make those nanogel dispersions using the carboxymethyl chitosan, get rid of the acetic acid. We tried that, and we got a DLS figure that showed us that we had multiple peaks of different sizes, right? So that we're not getting well-behaved formation of nanogels when we use the carboxymethyl chitosan. So what to do about that? And my student Zhenghao Zhai is the one who did all of this work, figured out, okay, well, we have all those carboxyl groups on carboxymethyl chitosan. What about if we just add a little bit of calcium and cross-link those carboxyl groups? So we have a second cross-linking mechanism. Imine formation, yes, but also calcium cross-linking of those carboxyl groups. And what he found was that as he added increasing amounts of calcium, and went from this multimodal distribution in DLS to a monomodal distribution, and that monomodal distribution being centered between 100 and 200 nanometers. So indeed, that worked very well. The dual cross-linking methodology allowed us to use Both polymers water soluble, no acetic acid to generate a nanogel dispersion. And we're pursuing the use of that nanogel dispersion for delivery of active molecules and for other applications. Okay, let's go to the third topic. I mentioned amorphous solid dispersion. So what the heck is that? This is a drug delivery thing. This is related to making poorly soluble drugs more soluble. So the pipelines of pharmaceutical companies these days are turning out these very hydrophobic drugs, and people like me that are organic chemists that get tasked with make me 100 analogs of that promising drug lead. Well, of course, we make 100 analogs, and we love to make crystalline analogs because they're easy to purify, and we organic chemists love to crystallize stuff, right? So what that generates is 100 analogs, all of which are quite hydrophobic because they bind tightly to hydrophobic pockets on protein targets, and also crystalline. So they've got two strikes against them with regard to water solubility. And sure enough, roughly 90% of the candidates that are coming out of pharmaceutical companies these days have got poor water solubility. And you and I are all water, mostly, so that's a problem. So amorphous solid dispersion developed as a way to address that. What is it? So we have this cartoon. We have our linear polymer over here. We have our crystalline drug over here. If we were to put that crystalline drug in the aqueous medium of your GI tract, only a few molecules would dissolve, and only a few of those would get through the epithelium that lines your GI tract into the bloodstream to get to the target organ. However, if we mix the drug and the polymer, we do that by extrusion or spray drying typically, we make essentially a miscible blend of the drug and the polymer. And because it's a miscible blend, there are drug molecules in between all of the drug molecules. There are polymer molecules in between all the drug molecules. Therefore, they can't crystallize. So you've got an amorphous drug, an amorphous solid dispersion of drug and polymer. If you put that into your aqueous medium of your GI tract, you create a supersaturated solution of that drug, supersaturated to an extent of 10 or 100 times versus its thermodynamic solubility. And so this isn't something that's a lab phenomenon. This is something that's in 25 to 35, somewhere in there, marketed drug formulations that you can go down to your drugstore and get right now. But the polymers that are used to do this are all pulled off the shelf. There are no polymers that were designed for this purpose. Sorry, there are roughly 48 approved, FDA approved formulations, the US Food and Drug Administration approved formulations. A lot of them use a cellulosic polymer, hydroxypropyl methyl cellulose acetate succinate being the most popular of those polymers. The polymers are repurposed. HPMCAS often works well, but it's very complicated to make. It's very complicated to analyze. And there are issues. There are issues with fast crystallizing drugs, which many of the new ones are. There are issues with low potency drugs. Why low potency? If it's a low potency drug, you need a lot of it in the formulation, right? You need, instead of 10 milligrams, maybe you need 100 milligrams in the formulation, which means the ratio of drug to polymer is going to be higher. And that means there's less polymer to prevent drug crystallization. So those are problematic for current ASD systems. So we've been trying to design polymers to do this for some time, and we made a lot of progress at it. We've learned, among other things, that the design of these polymers to do this is really challenging. And what do they have to do? Well, they have to prevent the drug from migrating in the solid dosage form, even when it's humid out, even if the drug happens to be a plasticizer for the polymer. That formulation has to stay above, with a TG above ambient temperature, no matter how high the ambient temperature is. And that means that it has to be a very high TG polymer. The polymer's got to dissolve in water. That's going to be the mechanism for release of the drug, is dissolution of the polymer in water. So it's got to be water soluble, but at the same time, it's got to interact with this hydrophobic drug in the solid phase and in solution phase. So it's got to be an amphiphilic polymer, water soluble, but also with hydrophobic characteristics. It's got to dissolve at a rate similar to the rate of dissolution of the drug, because if it doesn't do that, it's going to lead to drug crystallization either in the solid phase or in the liquid phase. Polysaccharides are advantage for this kind of thing because they're inherently high TG. They're inherently low toxicity. Of course, we know they're renewable based, and we can modify them. We can modify them to make them amphiphilic to enhance the solubility. If we're smart about how we do it, all the components of that polysaccharide derivative are going to be endogenous or dietary. So we're not going to have to worry about them from a toxicity point of view. So like I said, we made a lot of progress at this. My former student, Yifan Dong, did some of this work that I've talked about before, where we used olefin cross-metathesis to make some fairly sophisticated derivatives of polysaccharides. This particular work that I'm showing here started again with hydroxypropyl cellulose, but we're doing something quite different. We're, first of all, alkylating it with an alkylating agent that's got a terminal olefin, right? So now we have these hydrophobic substituents with a terminal olefin. We can use that terminal olefin for olefin cross-metathesis, ruthenium catalyzed, this being an electron-rich olefin by the Grubbs terminology, acrylic acid being an electron-poor olefin by the Grubbs terminology, which is a perfect pairing for olefin cross-metathesis. And sure enough, this is a grad student's dream reaction. It happens in an hour or so. It happens at close to room temperature. The conversion's 100%. The yield is way up into the 90% range. We hydrogenate that product to make it more stable, and that gives us this carboxypentyl hydroxypropyl cellulose. So we made a whole range of these things. We made a whole big structure property study, and it turned out this one has both hydrophobicity and hydrophilicity, that amphiphilicity we said we needed, and that carboxyl group for specific interactions with the drug molecules. And sure enough, it was a great ASD polymer, maybe the best we've ever seen. So this is a drug called enzalutamide. Someone you care about happens to have prostate cancer. They might be taking this drug right now, but they're dealing with the fact, the formulation deals with the fact that it's got terrible water solubility, less than 2 micrograms per milliliter water solubility. So we got some nice results in vitro. We took those results to a pharmaceutical company. They did some in vivo testing for us with rats, and that's what I'm showing you here. It's in vivo testing of that carboxypentyl hydroxypropyl cellulose as an ASD polymer with this prostate cancer drug enzalutamide. And we're showing area under the curve for different formulations, area under the curve being a way to express the amount of drug that gets into the bloodstream of the rat after an oral administration of this amorphous solid dispersion form. So this is crystalline enzalutamide, no ASD at all. And you can see that it's got very poor bioavailability. Very little of it gets into the bloodstream of the rat. This is an amorphous solid dispersion with that most popular current cellulose derivative, HPMCAS, at a ratio of 10 drug to 90 polymer, which is typical of ASDs. And it didn't improve on crystalline enzalutamide at all. It didn't help whatsoever. And it's because enzalutamide is a fast crystallizer. I told you that class of drugs has problems with current ASD polymers, and sure enough, this ASD polymer did not help it at all. This is the formulation in lipid that's called Labrosol. So you dissolve enzalutamide in a lipid, you put it in a capsule, you administer the capsule to the rat. It helps a lot, helps the bioavailability quite a bit. This is an amorphous solid dispersion with our polymer, carboxypentyl hydroxypropyl cellulose. Again, at the same ratio of drug to polymer as the HPMCAS experiment. What you see is that the bioavailability is essentially 100%. Every molecule of enzalutamide you administered to the rat got into the rat's bloodstream. That's going to mean in a human, if you were able to translate those results to a human, that you could administer less drug, you'd have less side effects, you'd have less costs, you'd have less drug polluting the environment after it passed through the rat, all kinds of benefits, right? So essentially 100% bioavailability. So we've solved the fast crystallizing problem. Here's a formulation with that same polymer, our polymer, but now at a 1-to-1 ratio, polymer to drug. And it's not quite as good as 1-to-9 ratio, but almost, and it's significantly better than the commercial formulation. So we've solved both the problem of fast crystallization and the problem of low potency drug at the same time. Really a promising derivative. So it's a great polymer. It's a great ASD polymer. Like I said, it's the best one I've seen, but it's got a problem. To make it requires this ruthenium organometallic complex to do the olefin cross metathesis. And we want these polymers to get to patients. And the only way they're going to get to patients is to have somebody make them. And people aren't willing to make them if they face this complicated and difficult catalyst recycling process. So it's challenging. We wanted to figure out a polymer, knowing these structure property relationships as we now do as a result of all this work. Can we design a polymer that works just as well, that has the right properties, but it's simpler to make? And so the way we approach that, my student Stella Petrova, who's in a postdoc right now, she's recently graduated, approached the problem by a simple ring opening chemistry. Can we ring open succinic anhydride or glutaric anhydride with a cellulose ester starting material to make materials that, from our knowledge of structure property relationships, would have that right combination of hydrophobicity, hydrophilicity, and ability to prevent drug crystallization. And so that's what she embarked on. And she was able to develop conditions for this ring opening reaction that gave high conversions, that gave high degrees of substitution of the omega carboxy alkanoyl group, either succinate or glutarate, and gave her quite high yields. And the properties of those polymers were pretty encouraging, right? They're all amphiphiles. We said we needed amphiphiles. They're all very high TG polymers, up in the 160 to 175 degree range, way over the 100 centigrade target that we have for these polymers. They're in spite of being amphiphilic, they're quite water soluble in the range of 300 to 400 microgram per milliliter, which is on the range of 6 to 8 times what we think we have to prevent crystallization and release the drug. Conditions to make them are relatively mild, so we're getting high molecular weight polymers, and they're pretty simple molecules. It's very easy to determine degree of substitution and even position of substitution by proton NMR spectroscopy. So we're very happy about the characteristics of these polymers, but do they work? So I'm going to show you a couple of pieces of data. This is for a drug called felodipine. It's an anti-blood pressure drug, current drug. Again, terrible water solubility, 1 microgram per milliliter. In its case, roughly 20% bioavailability because of that low water solubility. And so we did a comparison. So this is making a supersaturated solution of felodipine in the presence of a little bit of one of our polymers and comparing versus 2 different grades of HPMCAS. And what you can see is that we see differences in the time that it takes for the felodipine to start to crystallize. And for the better polymers, that time is on the order of 400 minutes. That is to say, well over 6 hours. The time that a drug takes to get through the absorptive part of your GI tract is about 240 minutes. All right, so this is preventing the drug from crystallizing for a longer period of time than it could possibly be in the absorptive zone of your GI tract. And it's better than one particular grade of HPMCAS, not quite as good as the other. Let's look at a different kind of experiment. This is a release experiment. So we're going to take felodipine again, same drug, same terrible water solubility. We're going to make an amorphous solid dispersion. We're going to add that to simulated fluid, simulated GI tract fluid, and watch the release of the drug. So on the y-axis, we have drug concentration. On the x-axis, we have time. Remember that somewhere in the range of 240 minutes is how long it's actually going to be in the absorptive zone of the GI tract. And you see 2 dotted lines on each one of these curves. The lower one, the black one, hard to see colors, but it's always the lower one. This is the thermodynamic solubility of felodipine. If you put felodipine in water, this is how much would dissolve. The top one is the amorphous solubility of felodipine. So it's the maximum solubility you could get in a supersaturated solution of felodipine. That also is a thermodynamic parameter, right? So this is the maximum amorphous solubility you can possibly get. Okay, so these are the 2 grades of HPMCAS. amorphous solid dispersions of fluidipine. You put it in, get a burst, high concentration, but then within a relatively short period of time, it starts to crystallize, and therefore you lose soluble fluidipine. So it's helping, it's definitely helping, but it's not performing exactly the way you'd like it to. And these bottom curves, A through F, are different cellulose acetate glutarates, cellulose acetate propionate glutarates, different degrees of substitution. And one of the things that all of this work tells us is that these polymers are exquisitely sensitive to the degree of substitution and the type of other substituent that's on there. In other words, we can really design for performance. And what you see is all of them help. All of them give concentrations of fluidipine that are above the thermodynamic solubility. But look at polymer E down here, which is the cellulose acetate glutarate. What you're seeing is that the concentration rapidly gets up to the maximum possible supersaturation, and it stays there. It stays there for the entire time that this dosage form would be in the absorptive portion of the GI tract. In other words, it's as good as it can possibly be. The performance is as good as it possibly could be. It's a simple cellulose acetate glutarate. So the story's over, you would think, but not quite. One more little wrinkle, and that'll be done. One of the things we saw with cellulose acetate succinates occasionally, and with cellulose acetate glutarates pretty often, was gelation during the period where we were trying to make it. And we developed conditions to avoid that. I'll talk about that in a minute. But we want to know why. Why was this going on, and what kind of conditions we need to use to avoid it? We had previously done work on cellulose adipates. Adipic anhydride has a lot of strain energy in the ring. It tends to homopolymerize. We're able to prove in that work that the mechanism of gelation with adipic anhydride was the fact that you grew oligoadipic anhydride side chains off of cellulose. Another molecule of cellulose came along. If it reacted at the anhydride linkage closest to cellulose, that would cause a cross-link. So that's what happens with adipic anhydride, and of course we thought probably that's what's happening with these glutarates and succinates. But Stella did rheological studies, and just long story short, what she saw was that with strain, you could break the crosslinks. With strain, you can break the crosslinks. That means they're not covalent. That means that polyanhydride is not probably the mechanism. Covalent bonds is not the mechanism of gelation here. We were able, fortunately, to work with a great group at Brandeis University on solid phase C13 examination of these hydrogels. And this is a solid phase C13 spectrum here. The quality of these things has gotten a lot better in recent years, and it gave us a tremendous amount of information. And one of the things it told us was that, of course, cellulose glutarates should have the same number of carboxyl carbonyls and ester carbonyls. There was an excess of ester over carboxyls in this spectrum. So that was telling us something. We weren't sure what. Also, as we looked at the carbonyl area in the C13 spectrum, down here around 170, where the anhydride carbonyls should be, there aren't any, right? So we know from that this anhydride cross-linking is not going on. The excess of ester bonds means that probably what's happening is ultimately we're getting ester cross-links. We're getting hydroxyl on one chain reacting with a carboxyl on another chain to make ester cross-links. And so what we saw, we think, is initial formation of non-covalent cross-links. Those are the initial gels that we've seen. Over time, those developing into ester cross-links, and that anhydride cross-linking is not happening with these cellulose glutarates. So we're able to take measures and prevent that cross-linking, simple measures and the synthesis process to be able to make these cellulose acetate glutarates successfully in high yield, and they are excellent amorphous solid dispersion polymers, and we're pursuing those now. Okay, so it just remains to thank this great group of students that's done the work that I showed you today. We love to collaborate. Klaus Schmidt-Rohr at Brandeis is the one that did that wonderful solid phase C13 spectroscopy. Lynn Taylor at Purdue has been our partner for all this amorphous solid dispersion work. She's a brilliant pharmaceutical scientist and physical chemist at Purdue University. My colleagues at Virginia Tech, Bob Moore and Lou Matson, really, really key to the block copolymer work that I talked about. And of course, we thank our funding agencies. And I thank you for your attention and happy to answer any questions.
Thank you a lot for a very nice presentation. It was covering so many interesting areas. I don't know. I can't see anything directly in the chat, so I take the opportunity to ask some questions in the beginning. You used the HPC. How sometimes the HPC changes, the side chains, the hydroxypropyl groups can chaining differently. Have you control over that, or by doing your own HPC, or do you buy them and have a distribution of HPC links?
Yeah, we haven't done that with HPC. In the paper we talked about that hydrogel formation. We talked both about HPC and also about hydroxypropyl dextran. And we used hydroxypropyl dextran because, as humans, we don't have cellulase enzymes in us. So if you're thinking about the kind of application that I talked about, where you're going to inject a hydrogel that contains drugs and treat an ailment in a human, you really shouldn't do that with hydroxypropyl cellulose. On the other hand, dextran is very easily cleared, and people get administered dextran intravenously in hospitals all the time, right? It's easily cleared from the human body. So we did make hydroxypropyl dextran. It's easy to do. You can control the degree of substitution and the molar substitution, and of course that controls the properties of your hydrogel. We're able to make materials that mimicked the physical properties of different human tissues, depending on how many ketones we put in by oxidation.
Okay, so but did you measure the lengths also, or was it more estimated?
We were able through NMR spectroscopy and through derivatizing the chain ends of the hydroxypropyl groups to get both MS and DS, and that's in the publications.
Thank you. Ulrika, do you have any questions?
Thank you. This was a wonderful lecture. Thank you so much. It's super interesting, all of it. So I have several questions actually, but I don't want to take all the spotlight from the rest of the audience, but I can start. So I was interested in
Ulrika, it's something strange with your mic. Maybe you can
really? You can hear me?
I can hear
a little garbled. Yeah.
But we can hear you.
Okay, good. Thank you. I'll try to be really clear. So you have this reductive animation to make a cross-link, and then you realize that the second cross-linking with ionic charged calcium would help in keeping the gel together. So I was just wondering, if you then put that in, say, saline solution, do you have ion exchange and you lose the secondary cross-linking by ion exchange sodium with calcium? Because that's known to happen for some of these ionically cross-linked receptors.
Yeah, I would imagine that those crosslinks would not necessarily last forever in a physiological environment, because the calciums, of course, could ion exchange out for monovalent ions. So I think that's absolutely correct. They would not necessarily last forever. Of course, you wouldn't necessarily want them to last forever in certain environments, but that is something you had to face. And let me correct one thing you said, that the cross-linking is not by reductive amination, it's by imine formation. We can do reductive amination. We can add for example, glycine and aminohexanoic acid permanently by reductive amination. But the cross-linking to make the hydrogels is not by reductive amination, it's by amination, by imine formation. And that's where the dynamic nature, that's where the self-healing comes from.
I see. I also have a question. You show many ways of basically tailor-making a polysaccharide, so you get really high selectivity, and you get exactly the groups that you need in this exact positions and so on, which is beautiful and I guess necessary for some of these really advanced applications like drug delivery matrices. If we think about having polysaccharides be a more bulk material that's going to replace big volumes of fossil-based polymers, I mean, what chemical modifications would be reasonable from a scalability perspective and also from, of course, a techno-economic perspective? What could we do basically that would be a true competitor to, say, polypropylene?
Yeah, it's a great question. I think if you look at the hydrogel formation chemistry that I talked about, that just involves bleach oxidation, it's very, very practical, right? It's selective, but it's also very practical. If you look at the amorphous solid dispersion polymer, given that much more complicated polymers that are tougher to make are successful right now, I think it's simple and practical. If you look at the block copolymer chemistry, I would certainly agree that the chemistry that we did there, you're probably not going to scale up to make plastic trays, right? But what we were really trying to do, and I don't think anybody's done this before, is to make materials whose structure was very well known, so that we could probe structure property relationships and we could see what do you really need to get a good compatibilizer. And I know, you Ulrika and Ann-Christine and others have made grafted polysaccharides before by simpler chemistries. And we definitely have the question in our group. We want to ask the question, is it possible to make these block copolymers by these simpler chemistries and still get similar results? Right? There are simpler chemistries, there are more practical chemistries that you can use to make some of these block copolymers. They're not going to be as regia selective as the chemistries we use, but that may or may not matter for their performance. Right. So that's an absolutely critical question that we need to address.
Yeah, I agree with you there. You might not always need the selectivity that is necessary for some applications, and maybe we can sort of cut a few corners there.
It gave us the information we needed, which is what we needed at this stage. But to turn it into something that's really practical, we have to go beyond that.
So what can we learn from starch, you think? You pointed out quite early in your lecture that starch has, a thermoformability, I mean, unlike most any other polysaccharide. So what is the secret of starch?
Yeah, I wish I had the answer for you. I mean, I can be glib about it and talk about, we all know, right? It doesn't pack as well as cellulose does. Cellulose is, you can think about it as trying to package slinkies versus trying to package steel rods, right? You can put so many steel rods into a package that you're sending to somebody that you can't pick the package up anymore, right? They pack very nicely, and that's cellulose, obviously. Slinkies, on the other hand, don't pack so well. You make a mess, and that's amylose. Amylose is helical. It's kind of like a slinky, right? So that's certainly part of it. That the self-association of the polymers that make up starch is not as high as the self-association as the ones that make up cellulose. That's part of it. But why specifically starch is so good at it, and something like dextran, for example, isn't, I can't tell you. It's just it's not known by me anyway.
I wish it worked. Okay, thank you so much. Do we have other questions in the chat? Maybe Josephine, you have a good view of the chat window?
No, I haven't seen any questions from the audience today. Or you already answered them, perhaps.
It was a very clear presentation, so
Yeah, it's really, really nice presentation. So I think that we need to close this webinar then.
yeah, we're close to 2 o'clock here. So I suppose we need, even if I would like to ask more questions, but I think we should close it.
So thank you so much, our speaker, and thank you so much to everyone who has been listening. So this webinar, we will, oh, sorry.
I enjoyed it very much. Thank you all.
Thank you a lot. And I hope we can see each other soon.
Me too.
Did you close it off so we can talk?
No, everyone is still here. We can't really
Okay.
close the webinar. So
Okay.
I think
Okay.